So let's talk about DNA replication. This is one of my favorite topics in biology. DNA replication is the process by which we copy our DNA. And it occurs in the cell cycle, specifically in the S phase, before we split our nucleus in two. So if this is our DNA duplex, then in the DNA replication, we're going to separate our parental strands and use each parental strand as a template to synthesize new strands. And we're going to follow our base pairing rules, in which adenine will base pair with thymine and cytosine will base pair with guanine. So here we can see that in each new DNA duplex, we have one strand that was in the original parental strand. It's the dark blue strand. And then we're going to have one strand that is newly synthesized. Here it's the light blue strand. This phenomenon is called the semi-conservative model. And the semi-conservative model is one in which we keep one old strand and we synthesize one new strand. Now here I've drawn our two parental strands, and remember that DNA is always anti-parallel, so we have a 5' to 3' strand and a 3' to 5' strand. The circular bubble that has formed is going to be called the replication bubble. And at the center of the replication bubble, we're going to have our origin of replication, ORI. So this is where we first opened our DNA and started to synthesize our new strands. The ends of the bubble are going to be called the replication forks. So here we have our replication forks. And at the replication fork, we have an enzyme called helicase that is unwinding our DNA. And helicase is opening the DNA so that we can replicate it. So our DNA is being opened in both directions at the replication fork, which means that replication is happening in both directions. But as we open our DNA, we're causing strain on our DNA strands. So an enzyme called Topoisomerase will be located after helicase and will help relieve that strain. Also, as we open our DNA strands want to join back together because they're complementary. They're more stable when they're hydrogen bonded. But to prevent the strands from joining together, we will have proteins called single-stranded binding proteins, SSB, that are going to prevent the strands from coming back together. Now let's talk about what the actual replication process looks like. There's a couple things to remember. The first is that DNA is always read from 3' to 5' and synthesized from 5' to 3'. The next thing to remember is that DNA polymerase, which is the enzyme that is synthesizing the new strand, can only add nucleotides to an existing strand. That means that we will add RNA primers to the start of each of our new daughter strands. Let's now look at the left half of our diagram and ignore the right half for now. The top strand reads 5' to 3' from left to right. The bottom strand reads 3' to 5' from left to right. So the top strand will be a template for a new DNA strand, and the bottom strand will also be a template. Let's start with the top strand. The first question you should ask yourself is, where will my RNA primer be placed? Remember that the RNA primer will initiate the start of the daughter strand, and the daughter strand should always read 5' to 3'. It will be anti-parallel to our template strand. So another rule of thumb is always place the RNA primer next to the 3' end of the DNA template. So in this diagram, the RNA primer would be placed here because this is next to the 3' end of the template strand. Since the direction of replication is happening to the left, the strand will be synthesized like this. If we do the same for the bottom strand, we can place the RNA primer next to the 3' end of the DNA template strand, and since the DNA can only be synthesized in the 5' to 3' direction, the strand will move away from the replication fork. Then, when we open our DNA a little more, we will place our primer again and DNA polymerase will synthesize in the 5' to 3' direction. This is how replication happens. We can now do the same for the right side of our diagram, and we'll get a diagram that looks like this. The strand that is synthesized continuously is called the leading strand. On the left side of the diagram, that is the top strand. And on the right side of the diagram, it is the bottom strand. The strand with multiple primers and that is synthesized discontinuously in pieces is called the lagging strand. These pieces or fragments are also called Okazaki fragments. So once we finish this process for our entire DNA, an enzyme called DNA ligase will remove these RNA primers and glue the fragments together. Also, DNA polymerase is able to proofread itself as soon as it makes an error. So if it mistakenly places the wrong base, then it has the ability to self-correct. And that is replication for you.